Hi everyone, my name is Courtney.
My name's Hannah.
And today we will be discussing the enzyme human hyaluronidase 1 along with its biological and chemical properties and its practical real-life application. In the 3D photo on the right, you can see a 3D print of hyaluronidase 1, which was pulled from the bioinformatics website UniPro. So during this presentation, we're going to start by talking about some general information about our chosen enzyme hyaluronidase, more specifically hyaluronidase 1, such as what it is and its functions and what it does, and then move on to its structure. So there are many different types of hyaluronidase, and it can be found in multiple species, but we are going to specifically be talking about hyaluronidase 1 throughout the presentation. And later on, we'll be discussing the primary literature article that we chose, which discusses the effects of cancer immunotherapy when utilizing hyaluronidase. So a little bit about what hyaluronidase is. So hyaluronidase is an enzyme that is known to break down a substance in the body known as hyaluronic acid, which can also be called hyaluranin. Throughout the presentation, Hannah and I will probably be referring to those terms interchangeably because they mean the same thing. Hyaluronic acid specifically is an anionic polysaccharide used to carry cells to regenerate tissue. So simply put, hyaluronic acid or hyaluranin is a fluid known to have a jelly-like substance that helps moisten and protect tissue in joints throughout the body. And in general, the use of hyaluronidase specifically helps break down that hyaluronic acid, causing the jelly-like fluid to become more liquefied and connective tissue to become thinner, allowing an introduced substance or said drug to move more easily into tissue. And in more chemistry-like terms, hyaluronidase is an endoglycosidase that is able to break down hyaluronic acid into monosaccharides by cleaving its glycosidic bonds. So the vertebrate hyal enzymes are of glycosidase family 56, defined as endo-beta-acetyl hexosaminidases that utilize hydrolysis and catalysis of HA at the beta 1 to 4 glycosidic linkages. So as you can see on this slide, I have pictured two structure pictures. The one on the left was pulled from the Swiss model bioinformatics website, and the one on the right is pulled from a peer review resource published by the American Chemical Society. And mainly I'll be focusing on the image on the right due to its specificity. So within the structure, there are two closely associated domains represented by the light blue and the yellow colors. The light blue is a catalytic like domain that has a distorted beta alpha 8 barrel, and the yellow portion is an EGF or epidermal growth factor like domain. The EGF like domain is intact and is for alternative splice variants, while the catalytic domain is likely to be unfolded. You can also see the active site of the enzyme shown in the middle here by the label and arrow. So a little about where hyaluronidase is found. It is present in many of the major organs throughout the body. So this includes the liver, kidney, and heart. Hyaluronidase 1 acts as a major hyaluronidase in plasma and is activated at an acidic pH level. And also hyaluronidase 1 can be found and conserved in different species, not just humans. So this can mean chimpanzees, monkeys, dogs, and mice. So here you can see the condensed version of the cDNA sequence of human hyaluronidase 1, and this in turn gives rise to the protein. And on this slide, you can see the amino acid sequence for hyaluronidase 1 that is 435 residues long and has a mass of 48,368 Daltons, which was found using the UniPro bioinformatics website as well. So with the use of the antigenic peptide prediction, the accompanying graph was generated. The predictions of peptides shown throughout represents the presence of amino acid residues in known segmental isotopes of the enzyme. And then on to the primary resource article that we chose. It is titled Highly Enhanced Cancer Immunotherapy by Combining Nanovaccine with Hyaluronidase, and this article was published in 2018 in the Biomaterial peer-reviewed journal.
Some little background about our experiment is what is immunotherapy. So immunotherapy is a cancer therapy. It stimulates the boost and anti-tumor immune responses of the body, enhances the immunogenicity of the tumor cells, and that improves the tumor sensitivity to the effector cells to suppress and eliminate the malignant tumor cells. There are several types of immunotherapy. For example, there's oncolytic virus therapy, immune system modulators, cancer vaccines, and monoclonal antibodies. Specifically, this experiment is going to be using cancer vaccines. In summary, just to summarize the total experiment, so this experiment introduces tumor antigens into the body, and it's going to make the subject's own immune system specifically activated to induce effective immune responses for controlling and eliminating malignant tumor cells. A nanovaccine is developed in this experiment to induce the antigen-specific anti-tumor immune responses. And hyaluronidase is known to break down hyaluronic acid or hyaluronin, has a wide range of tumors that are observed to contain high levels of hyaluronic acid. So tumors are known to have high concentration levels of hyaluronic acid, and those increase the interstitial fluid pressure in tumors and penetration. So hyaluronidase is going to break those glycosidic bonds and allow the nanovaccine to suppress the tumor cells better. And moving on to the materials and methods of our article. So first, the nanovaccine was created using polyethylamine, ovalbumin, and cytosine phosphate guanine. So those are the three main components of this nanovaccine. And so once that was created, the zeta potential and particle size of the POC vaccine, otherwise known as the PEI, OVA, and CpG vaccine, were measured using the potential PI90 plus particle size analyzer. This allowed us to determine the concentrations needed to input into the mice. And for the third method, morphological characteristics were observed using transmission electron microscopy, and the bone marrow-derived dendritic cells were harvested from the mice and isolated for experimental use. For the fifth method, confocal laser scanning microscopy was used. These stain the cells and wash them with a phosphate buffered saline in order to see the results. And then in vivo, so meaning like they use the treatment in actual live mice, the B16 OVA cells were injected into the mice, and the POC nanovaccine was injected one time a week, as well as the hyaluronidase group was also injected 2 times a week. And then in order to quantify their results, histological and immunofluorescence staining was used. Moving on to the results of the main article experiment. The cellular uptake of the PEI-OVA17 nanovaccine was significantly more efficient than the nanovaccine without a component of one of the three evaluated using flow cytometry. So as you can see in the graph, in the very last column, the PCO nanovaccine was used, and it has a much higher mean fluorescent intensity than all the others that didn't include all three components of the nanovaccine. So in this other slide, this is when they used the confocal laser scanning microscopy to stain the cell neurons, stain the cell nucleuses. And as you can see in the figure below, the blue ones are the nucleuses, and the green ones are the nanovaccine. So in the last column on the right side, the PCO nanovaccine shows significantly more green than any of the other columns, although the PO nanovaccine does have some green in it. So overall, the PCO and the hyaluronidase group led to smaller tumors. It also led to tumors with an 11-fold lighter than untreated group. That means that the over here in figure E, you can see that the hyaluronidase and PCO nanovaccine treated tumor cells were a lot lighter than the tumor cells that were untreated with the nanovaccine. As well as in figure C, you can see that the tumors that were treated with the nanovaccine and the hyaluronidase were significantly smaller, shown in the arrow. And then to suppress the high aggressives of the B16 OVA tumors, the nanovaccine was also used. As a side note, no mice died during the treatment process, and that revealed good safety of the PCO nanovaccine and the hyaluronidase treatment. On the other hand, when the tumors were palpable, the hyaluronidase was intratumorally injected into the mice twice a week for a total of 4 times, which led to higher injection of the nanovaccine into the tumor cells. Okay, so in this slide, it says that CD8 T cells play a major role in tumor suppression. They're known to kill tumor cells. So the PC vaccine plus the hyaluronidase of CD8 led to higher concentrations of the CD8 T cells in the tumor tissues than the other treatments. The hyaluronidase was employed to promote the penetrability of the tumor tissues for facilitating the infiltration of the vaccine-generated tumor-specific T cells in the tumors. So as you can see in the figures to the left, the nanovaccine had significantly higher results than all of the other treated ones, even in different environments. And then the final results are going to be led by Courtney.
So as mentioned in the materials and methods section previously, histological and immunofluorescent methods were utilized to analyze the tumor growth after being introduced to the varying treatment groups. And overall, there were no pathological abnormalities, and histomorphology was normal. Also, overall, the nanovaccine treatment group that was accompanied by hyaluronidase had a significant beneficial impact on tumor suppression and increased amounts of antigen-specific CD8 T plus cells. And this gives way to better and more efficient strategies for tumor treatments and therapies. And moving forward, hyaluronidase is still newly being studied in regards to tumor treatment, and will require more time and effort before being utilized in human subjects as tumor treatment.